All right, so the session is being recorded. This is going to be the first lesson in chapter 14 going forward. All right. Some of the things that are covered in this chapter were also mentioned in chapter 1, the introductory chapter. All right. So when we think about the origins and evolution of life, we figure that the formation of the Earth 4.5 billion years ago, okay? And then eventually the Earth started to cool down. We started to form the oceans and the continents. Okay. And then we have over here, somewhere around 3.5 billion years ago, we have the first type of cell, correct? The first type of cell. And the first type of cell, we already know what it is, correct? This is like review because it's like chapter 1. The first type of cell showed up maybe 3.5 billion years ago, somewhere around that time. What kind of cell? Good. It was prokaryotic. Prokaryotic. Okay. And what else did we know about it besides being prokaryotic? Was it aerobic or anaerobic? Was it an aerobic prokaryote or an anaerobic prokaryote? Take a look at the graph here. Do you see this pink line, what this is? See the pink line? Read the graph. You think that the very first cell, way back here, do you think it was aerobic or do you think it was anaerobic? Anaerobic, good. Annalene says anaerobic, and it had to be anaerobic because how much oxygen was even available in the early Earth? You see a lot of oxygen? Look how long it took before oxygen really started to show up on the planet, especially in the atmosphere. Correct? So it had to be prokaryotic and it had to be anaerobic. Okay? Why it had to be anaerobic? Because there was very little available oxygen, and you can't be aerobic unless you have oxygen, correct? Right? So it was prokaryotic because simple precedes what? Complex, and it had to be anaerobic. Okay? And then look what happens. So right after here, after this first living cell, what starts to happen over here? The first photosynthetic cells. So the first photosynthetic cells, right? For those cells, and it's still, you could take a look at the whole thing here. Take a look at the whole thing. The first photosynthetic cells, was that a prokaryote or a eukaryote? The first photosynthetic cells, 50-50 chance. Prokaryote. How can you tell? I mean, actually, it even tells you, right? If you read everything here, prokaryotic. And I mean by photosynthetic, I mean what? Photosynthesis. So who figured out photosynthesis first? Prokaryotes, correct? There weren't even any really what? There probably really weren't any, very few eukaryotic cells even at this point. Because this happened soon after what? The first what? Prokaryotic cells. Some of them started to become what? Photosynthetic. They figured out how to capture solar energy, okay, and turn it into ATP energy. That was like remarkable. Right? At the same time, some of the anaerobic prokaryotes started to get larger, okay? They started to get larger, and we could have had somewhere back over there, we could have had some prokaryotic, some eukaryotic anaerobic cells that developed also. Okay. All right. So first prokaryotic cell had to be anaerobic. We could have gotten some eukaryotic somewhere back there, eukaryotic cells, so some of the bacteria got larger and started to what? Take their DNA and put it in a membrane-bound nucleus. But soon after the first types of cells showed up on the planet, some of those early prokaryotes figured out how to do photosynthesis, correct? Because you see where that is. The first type of photosynthesis, okay, and photosynthesis was the first membrane-based mechanism for generating cellular energy. Okay, so photosynthesis was the first membrane-based mechanism, and that's the name of this chapter. Membrane-based mechanisms for developing cellular energy. And photosynthesis was first. Okay, first photosynthesis was the very first membrane-based mechanism for generating cellular energy, and it was figured out by prokaryotes. Okay, photosynthesis developed before aerobic respiration. See, we don't appreciate that. Okay, we seem to think that aerobic respiration is so important to us, it must be very, very old. But photosynthesis had to happen first. And the very first type of photosynthesis was called cyclic photophosphorylation. Because actually, photophosphorylation, I don't have any room to write this though, but photophosphorylation means using solar energy and capturing solar energy in order to phosphorylate ADP into ATP. That's what photophosphorylation means. Photophosphorylation is what happens during photosynthesis. Okay, that's how ATP is made in photosynthesis, through photophosphorylation. And the very first type of photophosphorylation did not involve what's called photolysis. So when we teach photosynthesis, we tend to always talk about this process called photolysis that takes place. But the very first type of photosynthesis was called cyclic photophosphorylation. And it did not involve photolysis. It involved taking the same pair of electrons that chlorophyll had and using solar energy to raise a pair of electrons to a higher energy level, and then release those high energy electrons and let them drain down, and then bring them back and use the same pair of electrons again. Okay, so it was called cyclic photophosphorylation. But then soon after that, the first water splitting photosynthesis took place. Soon after that, the first water split, and that's when photolysis comes in. So notice the first photosynthetic cells are over here, and that was before this process that is very popular today in photosynthesis, known as photolysis. And we call that non-cyclic photophosphorylation. And again, all of this is prokaryotic. All of these are the prokaryotes. All of this is the prokaryotes. Okay? So once the photosynthetic prokaryotes figured that they can do photolysis to provide a continual source of electrons from the splitting of water, instead of recycling the same electrons from chlorophyll, they said, wait a minute here, I can use solar energy to split water. And when I split water, I'm going to get 2 hydrogen electrons, 2 hydrogen protons, and I'm going to get leftover oxygen gas. I don't want the oxygen gas, but boy, I could use water, which was so important in biology, right, on the planet and biosphere. I could use water as an unlimited source of what? Free electrons that I can now use and keep putting into my photosynthesis mechanism, not have to keep on recycling the same pairs of electrons. Okay? So therefore, it became a very efficient way of actually doing photosynthesis. To use water molecules and use solar energy to split the water molecules apart, rip it apart. Water doesn't want to be ripped apart. And we call that important process photolysis. And that was a very productive way of doing photosynthesis. And it also provided another endpoint for where those electrons could go. Those electrons, some of those electrons could go to NADP to form NADPH. So this so-called, when you started to do photolysis, this so-called non-cyclic photophosphorylation was really important. But one of the problems, and this was considered a problem back then, okay, of doing photolysis, is that when I split water apart, okay, one of the waste products of this non-cyclic photophosphorylation was now going to be oxygen gas. Okay? And so oxygen gas was being produced from the splitting of water right back here at that time. And of course, all of this was really even happening in the ocean. Most of the life that started in a water environment, started in the ocean. And so what would happen is that these photosynthetic microbes that was now doing what non-cyclic photophosphorylation and splitting water, they were generating oxygen gas in the ocean. And in the ocean, because it was the early Earth, the Earth's crust had a lot of available iron. And what happens when you have oxygen and iron together is that the oxygen oxidizes the iron and turns into iron oxide, turns into rust. So the first thing that had to happen is that all this oxygen that's being generated in the ocean is first being used up to change any available iron into iron oxide in the ocean. And then once we actually so-called did that to all the available iron in the Earth's crust, not in the core of the Earth, but in the Earth's crust, then the oxygen would start bubbling out of the ocean and start to accumulate, okay, in the what? In the atmosphere. So this is, look at how long it took, right? Look at how long it took from when this was happening. It was taking like 1 billion years of doing this, what? Photolysis, to do photosynthesis and generating this potentially toxic waste product, oxygen, to have all that oxygen. We weren't worried about it, or they weren't worried about it, because it was just being what? Used to oxidize iron, changing it into iron oxide. So oxygen gas was not accumulating in the atmosphere. And the oxygen gas wasn't even really accumulating back then, even in the ocean, because it was being turned into rust. But suddenly now, once we finished using up all the available iron, now the oxygen that was being produced, and that all these microbes were doing that, suddenly now starts very rapidly filling up in the atmosphere. And now we have a problem, because oxygen, okay, in the atmosphere would be extremely toxic. So therefore, all the anaerobic cells, because everybody was anaerobic at this point, all of them were anaerobic, all before here, everything was anaerobic. Oxygen was considered fully toxic. But some of the microbes figured out how to take something toxic, oxygen, and turn it into something useful, and figured out how to do oxidative phosphorylation, figured out how to do aerobic respiration. Okay? And these organisms, these prokaryotes again, who figured out how to do aerobic respiration, they were able to live in areas and thrive in areas where anaerobic cells could not. So this became a big boom, meaning that once they figured out how to do that, they just went wild and crazy, and they started reproducing like crazy in an oxygen environment where all the other guys on the left-hand side were not able to live and had to hide away from, because oxygen was toxic to them, because they were doing everything anaerobically. So these aerobic prokaryotes become widespread. And of course, once you actually had that taking place, right, then we started to have the origins of eukaryotic photosynthetic cells, right? And how did that even happen? An anaerobic what? An anaerobic cell was able to what? To engulf an aerobic bacteria and became an aerobic cell, and then was able to actually also engulf some of these these photosynthetic bacteria, and you got the origins of eukaryotic photosynthetic cells that took place, right? And then once we had that going on, the eukaryotes took off, and we moved on to what? Multicellular plants and animals, eventually the vertebrates, and then eventually us. In fact, when we look, this is scary actually, when we look at the history of the Earth, and even the history of life starting back over here, all of this time, we talk about the first vertebrates being something like 0.5 billion years ago, correct? The first vertebrates. Where the heck is the first Homo sapiens? The first Homo sapiens would be like right over here. We have really not been on the planet, okay, anywhere near as long as other life forms have been on this planet. It's almost scary to think about 3.5 billion years ago what life was like, what very, very early life was like, right? When humans are like right there. We're right next to where that arrow says present day. Correct? All right. All right. So it took 1 billion years after photolysis, right? 1 billion years after photolysis before the Earth's atmosphere started to become oxygen rich. Okay. And the reason for that is because of all the available iron that was present in the early Earth's crust, in the biosphere area of the early Earth. And every time oxygen was generated by these organisms that were doing photolysis in their photosynthesis, okay, it was just quickly absorbed with the iron and became iron oxide. We call this period of time, this billion years from when first we started to do photolysis until this point over here, right, from here to here, we call that period of time the rusting of the earth, the rusting of the earth. And what was responsible for the rusting of the earth was the photosynthetic organisms who started to do photolysis as part of their photosynthesis, generating for the first time oxygen gas as a waste product. There really wasn't much oxygen gas that came with the earth, right? There really wasn't. The early earth, there wasn't oxygen gas. That was a very unusual gas to even be present with the early earth. But look at how important it is now to biology and the survival of organisms on planet Earth right now. All right, so that was because we had to rust up the earth. We had to convert the available iron into iron oxide, which is rust. Okay, so most of what was being produced dissolved in the early oceans, right? Start of rapid oxygen, iron, what? Okay, iron in the oceans was being used up, turning into iron oxide. All right, before aerobic respiration, how did non-photosynthetic anaerobic cells obtain their cellular energy? So before we had aerobic respiration, and we do understand what that is, right? How did non-photosynthetic anaerobic cells, whether they were prokaryotes or whether they were the early anaerobic eukaryotes, how did they obtain cellular energy? And when I say cellular energy, what am I saying? 3 letters, right? How did they make their ATPs? So before there was aerobic respiration, how did they make the ATP though? How did they make it? What process were they using? The only process, glycolysis. Excellent, right? Glycolysis is believed to be the oldest energy yielding metabolic pathway, correct? And so we just finished glycolysis in chapter what? 13, correct? And we know all about glycolysis. Yes, it is the oldest, the first and oldest energy yielding anaerobic pathway. It's the first energy yielding pathway ever evolved. Okay? Glycolysis came first, then photosynthesis. Correct? So glycolysis first, then photosynthesis. And the 2 types of photosynthesis, so we would say glycolysis was first. And then we had what's called cyclic photophosphorylation. And then from cyclic photophosphorylation, this was using solar energy. Substrate level phosphorylation to make ATP, right? Glycolysis. This is using solar energy to make ATP. And then after cyclic photophosphorylation, we have what's called non-cyclic photophosphorylation. And this is usually the one that we always talk about and that we always teach through biology, this one. Even though plants can actually still do both, plants can do both cyclic and non-cyclic photophosphorylation, but when you do non-cyclic, this includes what? Photolysis. And photolysis generates oxygen gas as a waste product. Okay. And then after non-cyclic photophosphorylation took off, eventually the prokaryotes figured out how to do what? Aerobic respiration. And again, that was the prokaryotes that figured out how to do that. And then aerobic respiration became very popular in both the prokaryotes, and even the eukaryotes. But remember, the eukaryotes never figured out aerobic respiration on their own, correct? Aerobic respiration became very popular. Once we had aerobic respiration, okay, once the prokaryotes figured it out, it was very popular for the prokaryotes to be an aerobic prokaryote, right? You can make a lot of ATP on that, correct? You don't need solar energy, right? You just need oxygen. You could do oxidative phosphorylation. The eukaryotes never figured it out on their own because how did the eukaryotes acquire it? Endosymbiosis, correct? What does that mean? The eukaryotes, how did they figure out how to do aerobic respiration? They never figured it out on their own. What did they have to do? They had to engulf an aerobic prokaryote and then live symbiotically with it, correct? So remember, eukaryotic cells are actually pretty stupid. Even when we think about aerobic respiration that we do, it's not really the eukaryotic cell that's doing it, it's the mitochondria that's doing it for the eukaryotic cell. Same thing with photosynthesis. Eukaryotes never figured out photosynthesis. Photosynthesis was figured out by prokaryotes. And so the green plant cells that are doing it now, they're doing it because they engulfed the cyanobacteria, or an ancestor to our cyanobacteria. All right, so you got that. That's the kind of order. Glycolysis first. Then we did cyclic photophosphorylation, which is a membrane-based mechanism. And that's really photosynthesis, right? And then what? They figured out how to do non-cyclic photophosphorylation. Okay. So that had its advantages over just being able to do cyclic photophosphorylation. And the big problem with that was the first time that a whole bunch of oxygen gas was now being evolved on this planet as a result of the activity of these prokaryotes. Right, so glycolysis is believed to be the oldest, energy yielding metabolic pathway. Is glycolysis a pathway? This is a yes or no question. Is glycolysis a pathway that uses a membrane-based mechanism? Remember, what's the name of this chapter? Membrane-based mechanisms of generating energy, cellular energy, right? That's what chapter 14 is. When did we do when did we do glycolysis? Last chapter, correct? Right, so what do you think the answer to this is? Glycolysis a pathway that uses membrane-based mechanisms to obtain energy? So I'm hearing yes and no. So which is it? Shira, what do you think? It's no, no, no. No, it's no. Okay. Isn't that funny how I can get you on a 50-50 question, right? It's no. Glycolysis does not require any membrane material, correct? Glycolysis is a catalytic pathway that occurs where? It doesn't occur in a membrane, does it? Okay. Glycolysis doesn't, none of the workers of glycolysis are in a membrane, right? So glycolysis is not a membrane-based mechanism, okay, to obtain energy. So what is? I guess photosynthesis is, and so is what? Oxidative phosphorylation. Okay, oxidative phosphorylation and photosynthesis are the two membrane-based mechanisms that cells use to obtain energy. Got it? Those two. That's really what this chapter is about. Oxidative phosphorylation, remember, which is like part of stage 3 in chapter 13. Okay, and photosynthesis. I actually go light on photosynthesis. There's more detail in chapter 14 on photosynthesis than even what I expect you to know. right? When you read the chapter. Okay. I mean, I give you a lot, on photosynthesis, but I don't give you every little detail about photosynthesis. I focus more on oxidative phosphorylation. All right. So 50-50 chance. This is scary, right? On true, false, even. Correct. The answer is no. Glycolysis has nothing to do with needing a membrane to work. None of the players are in a membrane. They require a membrane in order to get their work done. All right, the first aerobic cells were prokaryotes. We said that 1,000 times now. Some prokaryotes evolved a membrane-based mechanism that used toxic oxygen, okay, to release energy during the oxidation of fuel molecules. And these prokaryotes thrived, right? So in other words, once you had the prokaryotes that were doing photolysis during photosynthesis and making all this oxygen that was now accumulating in the biosphere, okay, this became a problem because oxygen is potentially toxic. So these prokaryotes figured out how to take something dangerous, okay, and use it to do something good, right? So these prokaryotes were able to live in places in the biosphere. They had their niche. They were able to live in places in the biosphere where other organisms were not able to live. Okay. Right? They were able to live where non-aerobic, and when it comes to evolution and survival and success, it all happens to do, and they use that term, a niche that you have to find where whatever you do, you are best suited to live in that environment, right? Okay. All right. Eventually, the early anaerobic eukaryotes, the stupid cells, right? The eukaryotic cells, us, right? We acquired the same membrane-based mechanisms, which means what? Oxidative phosphorylation and photosynthesis for energy production. But how? Did they develop this on their own? Did they figure out on their own? Another no. No, what did they do? They engulfed the aerobic bacteria that figured out how to do oxidative phosphorylation, or they might have also engulfed another type of prokaryote called an ancestor to our cyanobacteria. The cyanobacteria are the major, I guess, group of bacteria that do photosynthesis, right? Cyanobacteria. Okay, they engulfed an aerobic bacteria or the cyanobacteria, and then they did the endosymbiosis thing, correct? Today, membrane-based mechanisms, and what are the two membrane-based mechanisms? Oxidative phosphorylation and photosynthesis, are the major ways most cells obtain their cellular energy. That means ATPs, right? Right? See, students come into me thinking that photosynthesis is all about making glucose. And actually, that's wrong. Photosynthesis was a membrane-based mechanism that allowed you to change solar energy and conserve it in the form of ATP energy. Okay? The making of glucose is something that you do after the fact because you have ATP. Because you have it. Okay? You don't use solar energy to make the glucose itself. You use the solar energy to make ATP, right? So really, photosynthesis is about knowing how to capture solar energy to make cellular energy. And then what that cell does with the cellular energy, if you're a photosynthetic cell, green plant cell, is that you actually have the workers that can then change carbon dioxide into glucose, but using ATP energy to do that, not using solar energy to do that. Okay. All right. These mechanisms can be divided into two stages. So this is now, pay attention, going to apply to whether we're talking about photosynthesis or whether we're talking about oxidative phosphorylation. Unbelievable how similar the mechanisms are, even though we think of them as being, photosynthesis, oxidative phosphorylation, we think about them being two separate things because one's taking place in a chloroplast, what's taking place in a mitochondria. We think, oh, they got nothing in common. Totally wrong. These two mechanisms can be divided into the two stages, and this is true of both of those mechanisms. Stage one, what happens? High energy electrons have to be generated. That's the first thing that has to start. There has to be a way of generating high energy electrons and then transferring them to a series of electron carriers. When we say, where is the electron transport chain, right? We say, oh, it's in the inner membrane of the mitochondria. But you know what? There's more than one electron transport chain. I usually call that one the electron transport chain. Where is the electron transport chain? Because that is the most important one to me and to other animals. Okay? But a chloroplast, when you ask a chloroplast, where is the electron transport chain? They'll go, oh, I have one of those in the thylakoid membrane. Okay? And then you go, well, you have an electron transport chain. It's not the same one, but it is a series of what? Carriers of electrons that are doing something, okay, in that membrane. Okay? And there's even other electron transport chains in biochemistry as well. Not as famous, though, as the electron transport chain that we have in the inner membrane of the mitochondria, and the electron transport chain the chloroplast has in the thylakoid membrane. All right? All right, so we have to first generate high energy electrons somehow, have to be generated. And then those high energy electrons will be transferred to a series of electron carriers. And the electrons will move. So here's my carriers. I'm going to have carriers. Carrier 1, carrier 2, carrier 3. And the electrons are going to come in, and then they're going to be passed from carrier to carrier. All right? However long it is, however long the chain is. But electrons come in, and what kind of electrons are they? High energy electrons come in, and they move through various carriers, right? And then eventually the electrons come out. Okay, so this is an electron transport chain. What happens? Electrons come in, and they come in as high energy electrons, and then they eventually come out. And when they come out as low energy electrons. And do the electrons go in? Do the electrons come out happy, or do the electrons come out sad? So the electrons come in as high energy electrons. They move through the carriers. Oh, good. Tashira, see, some of my real fans here, they've been listening to me, because who else besides Dr. Carol talks about electrons being happy or sad, correct? So are they happy when they come out, or are they sad? They're happy because why? Annalie or Tashira, since you both said happy, why are they happy when they come out as low energy electrons? Because electrons represent matter, and matter always wants to move to a lower energy level. So they go in angry, unhappy, and then they come out what? They come out happy, correct? Right? And so therefore they move through their electron carriers, and as they are moving through, they must be losing their energy, right? And of course, since you're moving electrons from one thing to another, we say that each step in the movement is a redox step. That when the electron comes into number one has been reduced. And then when the electron leaves number one has been oxidized, and number 2 has been reduced, correct? So each transfer represents what's known as a redox step, and each redox step releases energy. Each redox step releases energy. So the electrons that are high energy, as they keep moving through the chain, they progressively lose their energy. So each redox step has to represent a what? An oxidation reduction step. Reduction gains energy, right? You gain energy when you've been reduced, and you lose energy when you've been oxidized. But if this is going to take place and you're going to come out with less energy, that means that the amount of energy you released is greater than the amount of energy required in the redox step. Otherwise, this wouldn't happen. Okay? All right, so you got that? Each redox step is going to release energy. Proteins in the membrane capture some of the energy because proteins are workers. Proteins are smart. Proteins are like machines. Okay? And when you give what? When you give a machine, say, an electricity, right? Because maybe the electricity comes in, it can capture some of the energy of the electricity and transfer it into mechanical energy, correct? Because they're machines, they're smart, they know what to do. They know how to capture one form of energy and change it into another, correct? So proteins in the membrane can capture some of the energy released during the redox action and do it to do and capture it and use it to do work. Right? Just like a machine does. A machine does work. That's why we build machines, because they do work for us. Right? Okay, we give them energy and then they do some kind of work with that energy. They know how to capture the energy to do some kind of work. That's what these proteins are. They're like machines. And then the question is, all right, they're in an electron transport chain, correct? Keep that in mind. What kind of work do these proteins do? And this was actually a very big question when we were learning about this, in the research behind all this biochemistry and cell biology. That was actually a big question. What exactly are they doing with the energy? They know electrons come in as high energy electrons. They even know that they go out, and they go, what kind of work was done by these electron redox reactions? What kind of work was done? And then we take a look at the picture and what kind of work is going to be done. Very good. Proton pumping. So in stage 1, electron transport drives pumps that pump protons across the membrane. Remember, anything that's called a pump is active, is energy requiring. You can't pump something. You can't pump iron in a gym. You can't pump water from a well unless you actually expend energy, because pumping is always implied as being some kind of work that requires energy. So what are they pumping? They're pumping protons across the membrane. They're pumping protons to where they don't want to go. So let's see what happens. High energy electrons come in. Look at that. They go out at low energy electrons. The green protein here is capturing some of that energy. And what is the red work that it's doing? It's moving hydrogen protons from low concentration on one side of the membrane and pumping them out to high concentration. And they don't want to go, correct? All right? And you're pumping them from one place to another using the membrane as a barrier that those hydrogen protons are unable to diffuse back in, right? Because as we know, a phospholipid bilayer is particularly a barrier to anything that's charged. Even tiny hydrogen protons cannot diffuse across the phospholipid bilayer. Okay? So in stage 1, what do? Electron transport is used to pump protons across the membrane and set up a proton gradient across that membrane. And a proton gradient is an electrical chemical gradient. It's a double gradient. It's potentially, at least, a double gradient. Potentially a double gradient because why? Because not only is there going to be a concentration difference, but because whatever we're talking about being high-low concentration is also carrying a charge, right? So there's going to be a difference in charge across the membrane as well as a difference in concentration as a result of pumping protons, all right? Okay, membrane proteins are essential for this process. Right? You know how much I think about proteins, right? Because they're workers. They're smart. They're like machines. Okay? They do work. Right? You have to be smart to do work. You're like a machine. You're not just what? Mass. It's not just matter. You have to be able to do some kind of work. Without the membrane proteins that know how to capture the energy of electron transfer, what would become of all the energy released if you didn't have smart workers, machines that was able to capture the energy as the electrons are releasing it? The electrons are moving spontaneously from member to member and losing their energy. What would happen to the energy that's released if you didn't have these machines in the membrane that knew how to capture the energy and do some kind of work with it? What happened to it? What does the Queen say? It will be wasted. Excellent. It will be wasted energy. And what will it be wasted as? What kind of energy? What does the Queen say? Give me my what? Give me my heat energy. Excellent. The energy would be lost, wasted as heat energy. Okay. Here's some more multiple choice questions that I'm putting in here to make you think about it as I ask it. You know, if you're reading the slide, you probably already know the answer because you're looking right at it. I don't care if you're even doing that. I mean, that's better than doing nothing, or better than just folding laundry, okay, while I'm talking. What is the ultimate source of the protons that are being pumped across the membrane? You just saw pictures of the protons being pumped across the membrane. Where, okay, in the cell, where in the cell would we be getting these protons from? And again, this is kind of like, well, I don't know, where are we going to get them from? Right? Where do we get them from? Okay, and the answer to that, where are these protons that are being constantly pumped across? Where are they coming from? They're coming from water molecules. Because what do we know about water, even though it's a covalent compound, right? Even though it's a covalent compound, what do we know that water has the ability to do? To dissociate, correct? Right? Isn't that true? To a very small extent, right? No, it's like nothing. How much water actually does that? 10 to the minus 7, right? 10 to the minus 7. That's like nothing, correct? But we know water can do this, right? And we know that water wants to have an equilibrium about doing this, correct? Isn't that true? Water has an equilibrium at a given temperature. By how much, what, hydrogen protons and hydroxide ions we actually would want to generate from the dissociation of water. And how can I make this happen faster? How can I push this to the right? I could push this to the right by adding more water. Well, that's not going to work. Right? I can't just add more water to the body. But how can I actually get this to be pushed or shift to the right? Remember shifting, equilibrium and stuff like that? Shifts to the left, shift to the right. How can I get more water molecules to continue to do this? By removing one of them, by sending it across a what? A membrane barrier. If I send it across a membrane barrier, I disturb the equilibrium on the other side of that membrane. And it will help force another water molecule to dissociate to replace the hydrogen proton that was lost, correct? That's what happens in an equilibrium situation. And then I just what? I pump more of them out there. And the more that I keep pumping, the more I can still get from the breakdown of another water molecule, dissociation of another water molecule into its ion. And of course, since I'm only pumping out one thing, I'm only pumping out the hydrogen protons, what's going to happen to the concentration of the hydroxide ion on the other side? The concentration of the hydrogen ion will go up over here. And what will happen to the concentration of the hydroxide ion on the other side, who's not being pumped across? Concentration of this will go up over here, correct? Right? So when you pump protons across a membrane, not only do you get an electrical difference because you're putting out a whole bunch of what? positive charges on this side of the membrane, and therefore accumulating more negative charges on the other side of the membrane, you get a voltage gradient and you get a concentration gradient with the hydrogen protons. So this is a voltage gradient. Right? And of course, the fact that you have a high amount of hydrogen protons on one side and the other, you have a concentration gradient as well. And you know what else is weird about when you're pumping protons? When you have a concentration gradient of hydrogen ions that's different on one side of the membrane composed to the other other thing you have is a pH difference. Because I could have sworn pH is what? Is related to the concentration of the what? Hydrogen ions. And suddenly now on one side of the membrane, I have more hydrogen ions than I have on the other side of the membrane. So which side of that membrane, the top side here or the bottom side, is going to be more acidic? And which one's going to be more alkaline? Which side of the membrane, the way I drew it, is it going to be the top side or the bottom side? It's going to be a pH that's going to be more acidic. Well, I guess where the hydrogen protons are going is going to be where it's more acidic. More acidic. Out here. More alkaline. Oh, no. No, no. I hate when this happens because I don't know how to get back to it. Oh, I kind of got back to it, I think. Let's go come back over here, though. Come here. Okay. More alkaline over here. All right. So, wow, when you're pumping protons across a membrane, there's lots of effects of that. When you get a proton gradient, because a proton gradient is going to be a voltage gradient, it's going to be a concentration gradient, and because we're talking hydrogen ions, it's also going to be a pH gradient, because that's what we talk about with pH, difference in what hydrogen ion concentration. So that's like wild. All right. All right, so stage 2, what happens in stage 2? A proton gradient has been made, and that proton gradient is like a battery. Proton gradient, battery. Stored energy. Okay, here I have pissed off, what? Pissed off, angry, A-N-G-R-Y, hydrogen protons over there on that side. They're pissed off and angry. Why are they pissed off and angry? Because what? They're with a high concentration of themselves and they want to go back to the other side, correct? So they're angry. There's a big crowd of hydrogen protons that have now been pumped out against their will, and they're all on top of each other, yelling and screaming and pissed off that they are now what? Being pumped out to an area of high concentration. And their natural thing is that they want to go back to an area of low concentration. And they're angry. They want to get back in, but they can't get back in. Why not? Because a phospholipid bilayer is a fantastic barrier to the passive diffusion of anything charged, including tiny hydrogen protons, cannot diffuse back into the other side of the membrane. So they get angry, they get crowded. Almost the same way that years ago, there used to be over in, I think it was the Valley Stream Walmart, Black Friday, when they used to have all the sales. And I think it was Walmart over in Valley Stream, over near me, actually, where I live on Long Island, okay, right over the Queens border, that actually was having their, Black Friday sale. And they were like, they had like 5 big screen TV sets for an unbelievably low price. You had to be there and when the doors opened at 4 o'clock in the morning or whatever crazy time it was that the doors were going to open. So people started lining up midnight, okay? And they stayed outside in the cold and the wet, whatever the terrible weather was, right? And then as they got there, if you got there at midnight, you were right near the door. And now as other people are coming, they want to be near the door, right? And everybody starts pushing and pushing and pushing, trying to get close, right? And they start getting to be an angry crowd because people are trying to cut. And they're trying to get closer and closer. And you're going, I've been here since midnight. What are you doing? You just got here. Hey, get back in the back of the line, correct? A lot of anger going on for Black Friday, sales, correct? And then all of a sudden, we're getting really close to the doors opening. And then there's a big push. Everybody starts pushing close to the door now, okay? Because there's going to be They're going to open the door. The poor little people who are working there are going to have to open the door. And there might be 500 people that are outside that door who want to get in first and run to wherever they're, big screen TV or wherever they're selling it to try to get a hold of it or whatever they came for. And then what happened was when the employee opened up the door, the employee actually got trampled on and other people got trampled on with everybody running in. And of course, when you run in, you're so angry. But then if you got to be the first couple of people in and you got in, suddenly all of your anger from being outside since midnight all dissipated as you went from high concentration outside and you got in and the store was empty, low concentration, and you got in. And all the camera people would be on the inside of the store showing all the happy people who got in first. And it's the exact same thing for these hydrogen protons. They're pissed off and angry, banging on the membrane door saying, let me in, let me in. I have all this pent up energy, all this pent anger. And then all of a sudden, this guy over here, who's not part of the electron transport chain, who did not pump any protons, he says, oh, here, you can't get through the membrane? That's too bad. Let me help you. Let me help you get through. I have a little door. I have a channel that you can come through. And when you come through and you release your energy, because you're going to be so happy going from high concentration to low concentration, this will be spontaneous and exergonic for you. When you come through my little channel, okay, when you come through my little, it's almost like a revolving door thing. Okay? It's like a machine, amazingly. When you come through, okay, and you release your energy, I'm going to use this energy to do some kind of work. And what kind of work does he do on the other side of the membrane as they are coming back through? He synthesizes who? ATP, correct? And he's not a member of the electron transport chain. In fact, he's doing the exact opposite. This guy is working. His job is what? He's pumping. And this guy, and he's making the proton gradient. This guy is draining the proton gradient. So he's doing pumping work. He's pumping work to make the proton gradient, and he's draining. Draining the gradient. And then this guy yells at him over here and he goes, wait a minute, I'm trying to make a proton gradient, stop draining it. But of course, what? So they're at odds with each other as to what kind of activities they're doing. But in the long run, we're happy because why? When he's draining that battery and he's letting them back in, he's capturing some of the energy they release to do some work of their own. And of course, that work is what? ATP synthesis. Right? So it's an interesting dynamics of what's going on in stage 2, right? So some of the energy released when the protons are allowed to move down their electrical chemical gradient is captured by this worker, this guy, and used to drive ATP synthesis. And who is that famous enzyme? This is an enzyme because he's phosphorylating ADP into ATP. Who is that enzyme? ATP synthase. And where is ATP synthase then located? He's a membrane, he's an embedded membrane. He's a transmembrane protein. He's a big, very famous transmembrane protein. Okay? And where do we have that membrane protein? It's in the inner membrane of the mitochondria. And where do we have the members of the electron transport chain? Same membrane, the inner membrane of the mitochondria. Okay? And of course, this is present not only in the inner membrane of the mitochondria, but the thylakoid membrane has its own electron transport chain in the membrane. And the membrane, the thylakoid membrane, also has ATP synthase in the membrane, the thylakoid membrane. And of course, that electron transport chain and the ATP synthase in the thylakoid membrane is really all part of what? Stage 2 here, stage 1 and stage 2 that takes place during photosynthesis. Okay? So in other words, this is not something unique to only the inner membrane, the mitochondria. This is the same thing that's happening during photophosphorylation, okay, in photosynthesis. Okay, the light-dependent reactions of photosynthesis. All right, so what is this interesting sequence of events called? It has a crazy name. The sequence of events that I'm just describing, that stage 1 to stage 2 sequence of events, is referred to as chemiosmotic coupling. And it's the process that couples 3 activities together. The 3 activities would be electron transport. Okay, so electron transport leads to proton pumping. And then proton pumping leads to ATP synthesis. Okay, so the electron transport chain, what does he do? He does work. And what kind of work does he do? Proton pumping, correct? Right? And then the proton gradient, okay, proton pumping sets up a proton gradient. Proton gradient. And that proton gradient is then stored energy as a battery, right? Battery. Stored energy. Okay, so the electron transport chain does work, right? Where does the electron transport chain get the energy from to do the work? Where does the electron transport chain get energy from to do its work? What comes into the electron transport chain is high energy electrons, and what comes out of the electron transport chain is low energy electrons. So where does the electron transport chain get the energy from? From the redox reactions of the electrons moving through. You get it? And then the electron transport chain has to do some sort of work. What kind of work does it do? It does the proton pumping. Proton pumping sets up a proton gradient, which represents stored energy, which is going to what? Be responsible for what? ATP synthesis via an enzyme called ATP synthase. You get it? So that's stored energy that the electron transport chain was responsible for is then going to be used or drained, okay, to make ATP synthesis. So that stored energy is going to be used to do work. And the work that's going to be done is ATP synthesis. And who's going to do that work? A very famous enzyme called ATP synthase. Okay. Is chemiosmotic coupling used in photosynthesis or aerobic respiration? Trick question. Is chemiosmotic coupling, this concept of having an electron transport chain, okay, both, correct. Most students would what? Guess one. Most students would just guess aerobic respiration because they really could care less about photosynthesis. This mechanism is present in both, the exact same mechanism, even though the electron transport chain is slightly different players in photosynthesis, different named players in photosynthesis than they are named players in the mitochondria. But it's actually the same ATP synthase protein that does the ATP. It's the same enzyme, same named enzyme. Okay? But the names of the electron transport chain members are a little bit different in photosynthesis than they are in oxidative phosphorylation. So it's both. That's the important thing there. Both. So I think you have to know the correct order of chemiosmotic coupling. What has to come first? The electron transport chain has to do its work first. Second step, forming the what? Proton gradient, right? And the next step leads to ATP synthesis, correct? So what would happen if the electron transport chain stops working? Then there'll be no proton pumping, and then there'll be no what? ATP synthesis. You get it? So if you stop the electron transport chain, if that doesn't work, then you can't do the second step, and then you can't do the third step, correct? Right? What happens if you had an electron transport chain and you pump the protons, but then the protons were able to sneak back in? Suppose they were able to sneak back in on their own. Something was wrong with the membrane. Suppose the membrane was damaged, then it's not going to work, right? You're not going to be able to get the next step because the proton pumping has to be able to generate this proton gradient, because that is the source of the energy needed by this enzyme. This enzyme cannot use the energy of the redox reaction above. This enzyme gets its energy from the proton gradient. And the proton gradient is a what? Is the end result of the electron transport chain doing its work. Okay? This actually was only figured out, I'd say maybe in the 1960s. They actually really figured this out. So this is not one of these biome things that they've known for like, over 100 years or something. It was really not until about the 60s, maybe even the 70s, that this was really fully worked out, this chemiosmotic coupling thing. Okay? All right, sources of electrons. So we know what the source of the protons are for proton pumping. Who remembers? Who remembers out of who's here? My favorite students. Who remembers? The ones that came today. How nice of you. Who remembers what the source of the protons were for proton pumping? Nobody remembers? I'm not making an impression? No, the source of the protons. Protons. You know what protons look like? The source of the protons. What was the source of the No. How could redox? Redox deals with electrons, right? Remember? Redox reaction only talks about electrons. What was the source of the protons? No. What do you mean hydrogen? This hydrogen is a proton. Okay. Rose Laura. I don't know why I'm having so much trouble with your name. Rose Laura. Okay. Water. Very good. The source of the protons was from water. Correct? Right? So what is the source of the protons for proton pumping? It's an unlimited supply of protons that can be pumped, because there's an unlimited supply of water in the cell, correct? Really? I think of how much water is there. So the source of the protons for proton pumping comes from water. The dissociation of water, right? Do I have to write it again? Remember the dissociation of water that you learned in chemistry and you thought it didn't have anything to do with biology? Okay? So therefore, water is the source of the protons that are then pumped across the membrane. And only the protons are pumped. And when you keep on removing the hydrogen protons, it causes another water molecule to want to associate to replace it. That's chemical equilibrium for you, correct? All right, so the source of the protons, we said, is going to actually be the dissociation of water. Now we're asking, what is the source of the electrons? The electrons aren't pumped, are they? Do we pump any electrons across a membrane? Yes or no? Do we actually pump protons, or do we pump electrons? Are the electrons moving across the membrane? No. The electrons are in the membrane, moving from member to member, but they never cross from one side to the other. So electrons are not being pumped or pushed or allowed across the membrane. Only the protons are being pumped. There's no such thing as the electron being pumped. So what is the source of the electrons that go into the electron transport chain, right? That's what the electrons do. They go into the electron transport chain. So in aerobic respiration, what is the source of those high energy electrons? You know aerobic respiration, right? Chapter 13. Aerobic respiration, chapter 13. Where do we get the electrons, those high energy electrons from? that we're going to send to the electron transfer chain? No, chapter 13, like it's going to be on the test this weekend. In aerobic respiration, right? We know we have to have high energy electrons. Chapter 13 that are generated, correct? That go to the electron transport chain. So what is the source? Where do those high energy electrons originally come from? Citric acid cycle. Good, we're getting warm. Citric acid cycle. What's happening in the citric acid cycle though? Where do the electrons really come from? No, I mean, you're good with the citric acid cycle. I think exactly tell me what electrons have to come from some kind of matter, right? Electrons are matter. They have to come from somebody. So where do the electrons, these high energy electrons that NADH has, right? NADH has the high energy electrons. Where did they really come from? What are they coming from? No one? That's a good question, actually, to understanding chapter 13. They're coming from oxidation of fuel molecules, right? So really, the electrons are coming from like the oxidation of glucose. No, where? When I say where, I don't mean like in where are they located. I want to know where they came from. Okay, they might have come from the cytosol, but they come from the cytosol because the electrons originally came from the oxidation of the carbons of glucose. Remember, glycolysis is an oxidizing pathway. Oxidation always releases energy. Some of the energy gets conserved in the form of high energy electrons. Yes? Okay. And where are these electrons originally from? I had to pull the electrons away from carbon, right? Because what are we doing in glycolysis and Krebs cycle? We're oxidizing, we're removing electrons from fuel molecules. So we're removing electrons from glucose in glycolysis. We're removing electrons from pyruvate dehydrogenase. We're removing electrons from acetyl-CoA during the Krebs cycle. And when we remove electrons, it means we oxidize that chemical and we release energy. And we keep some of that energy to make those electrons that we took away high energy electrons, right? Because that's what we want to make. We want to make high energy electrons in the Krebs cycle. But when we say we want to oxidize the acetyl-CoA, we don't really want to oxidize the acetyl-CoA. We get carbon dioxide. What are we going to do with that? It's a waste product. We want to oxidize the acetyl-CoA to generate high energy electrons. That's why we oxidize it. Not because we want to make carbon dioxide. Carbon dioxide is no good. It's a waste product. Get rid of it. But what we keep is the high energy electrons. And then where do we send the high energy electrons? Where else should we send the high energy electron? To the electron transport chain. What is the source of the electrons? So I'm going to stress again the source of these high energy electrons, because low energy electrons will not go to chemiosmotic coupling. High energy electrons, right? High energy electrons in chemiosmotic coupling. During aerobic respiration, it comes from the oxidation of the fuel molecules. That means it's coming from glucose being oxidized. Pyruvate, remember we talked about this. This was last chapter, being oxidized. Okay, acetyl-CoA being oxidized. Fatty acids being oxidized. Okay, even amino acids that eventually get oxidized. Those are all these fuel molecules here that we oxidize during aerobic respiration, okay, generate high energy electrons that can be used for chemiosmotic coupling. So you got that? Okay, from photosynthesis, because remember, chemiosmotic coupling takes place during aerobic respiration and during photosynthesis, correct? So where do we get the high energy electrons from in photosynthesis? From activated chlorophyll molecules. That's the important role. Remember, what can chlorophyll do? Chlorophyll can capture solar energy. And how does it capture the solar energy? Some of the electrons in the chlorophyll molecule get activated by the solar energy and are raised by the solar energy to a higher energy level. That's the way the chlorophyll molecule works. When you put it in the sun, certain wavelengths of light, particularly red, believe it or not, in the chlorophyll molecule, also blue, those wavelengths of light excite pairs of electrons, or a pair of electrons in the chlorophyll molecule, and raise those electrons to a higher energy level. And then what does the chlorophyll molecule do with those high energy electrons that have been raised to a higher energy level by the sun? It throws it into an electron transport chain to start off the chemiosmosis process. So activated chlorophyll molecules, who can get them ultimately from water, right? So in other words, if I was the chlorophyll molecule here, so this is chlorophyll. Okay, so the chlorophyll molecule is going to have a pair of electrons in its structure, okay? And then it's going to have solar energy that's going to excite the pair of electrons to a higher energy level. And then once you get into that higher energy level, you go into the ETC, the electron transport chain. And then what? The energy gets released and it can come right back. So they come in, they're high energy electrons. They go into the electron transport chain and they come out as low energy electrons, right? Because isn't that what happens? You go from high energy electrons, you go into the photosynthesis electrons, and you come out as low energy electrons. You come back to the chlorophyll molecule, and then you hang out in the sun again. And the same pair of electrons gets raised to a higher energy level, goes into the electron transport chain, comes out in the low energy level. And you go back again. This is called cyclic. That's cyclic photophosphorylation, because you keep cycling the same pair of electrons. You use the sun to raise them to a higher energy level, put them into the electron transport chain, let them do the chemiosmosis thing, move to a lower energy level, pump the protons, do all that stuff, and then those low energy levels come back to the chlorophyll molecule, and you do it all over again, okay? And then if you didn't want to do cyclic ones, then what you could do is you could take water, break it down into a pair of electrons, pair of protons, and oxygen gas, and you could take these low energy electrons and keep giving it to the chlorophyll molecule. So every time the chlorophyll sends up a pair of electrons to the electron transport chain, we wouldn't have to wait for them to come back. We could split another water molecule, do it again, split another water molecule, do it again. So it's a more effective way of actually running what? Photosynthesis. And that's called non-cyclic photophosphorylation. Again, you know, all of this, you can read about it in the textbook too, right? So it's always fun to read about these things in the textbook. Or just listen to like videos, re-listen to this, all right? So where do we get the electrons from? What is the source of the high energy electrons in photosynthesis? Activated chlorophyll molecules, which ultimately could have come from water via photolysis, okay? So the idea is that they're not high energy electrons until chlorophyll activates them, okay? So when they come right out of the water molecule, they're low energy, but then chlorophyll will activate those electrons, and then they can go into chemiosmotic coupling. And this is its own area if you were taking microbiology and you were really studying some of the special biochemistry of some special bacteria, such as the chemisynthetic ones. We're not going to spend a lot of time talking about that, but this is actually the third place where we have, third situation really, where we have chemiosmotic coupling types of reactions membrane-based reactions. And these are the chemisynthetic bacteria that might leap in the ocean. And they also can actually do chemiosmotic coupling. But what they use is they use certain inorganic molecules like hydrogen gas, iron, or sulfur. So that's its own biochemistry. They mention it in the chapter a little bit, but they don't go into detail about it. So I'm thrilled if you know these two, more so than even knowing what happens with the chemisynthetic bacteria, which again they're very often living in inhospitable places as far as I'm concerned, right? Like maybe deep down in the ocean, okay, where we have some of those chemisynthetic bacteria. All right, dinitrophenol, DNP, okay, dinitrophenol, okay, is something that interferes with chemiosmotic coupling. One of the things that when they discovered chemiosmotic coupling, or they realized what was going on, they were looking for things that could interfere with chemiosmotic coupling. And all the things that interfere with chemiosmotic coupling would be toxic. Okay, so these are toxic chemicals that interfere with some step of chemiosmotic coupling. Either it interferes and kills the electron transport chain. So there's like lots of things that can do that, like carbon monoxide. You know about carbon monoxide? Carbon monoxide interferes with chemiosmotic coupling because it actually interferes with the last step of the electron transport chain, right? Cyanide interferes with the electron transport chain, okay? And there's this other interesting chemical called dinitrophenol that interferes with chemiosmotic coupling, but doesn't go after the electron transport chain. It actually goes there a middle step in chemiosmotic coupling. What dinitrophenol does is that it's a chemical that makes the inner membrane of the mitochondria permeable to hydrogen protons. Remember what we said about the membrane? The membrane is really what? Very much a barrier against the diffusion of charged particles, even the protons. So if you get exposed to dinitrophenol, dinitrophenol is going to poke like holes in the inner membrane of the mitochondria. So that's the only thing it does. It doesn't do anything to the electron transport chain. So electrons will end up coming to the electron transport chain and moving through the electron transport chain, and protons will be pumped across the membrane. But then instead of forming a proton gradient, they leak back. So we don't form the proton gradient. The proton gradient doesn't form, okay? And if the proton gradient doesn't form, what can't work? Then we can't do ATP synthesis. So you're interfering with the middle step in chemiosmosis, because even though you're pumping the protons, the pumping of the protons does not lead to the formation of that stored energy. Remember, this is stored energy, the type of energy that the enzyme ATP synthase needs, okay? All right, so they leak back in across the membrane. But when they leak back in, aren't they still happy that they got back? And that means when they leak back in through leaking, energy is released. Energy is still released, but what happens? The energy is not what? Conserved, right? So they come back in, they leak back in, and they get what they want, but they're not coming in through what? ATP synthase, right? They're not coming in over here. And so what's going to happen when energy is going to be released? So the electron transport chain captures the energy from the high energy electrons, and then it what? Does the work, it pumps the protons out, but then the protons just laugh and they leak back in, correct? So you never form the proton gradient. But when they leak back in, they release their energy going from high concentration to low concentration. And when they release their energy, how does it come out then, since it can't be conserved? What happens to the energy that's going to be released if it can't be conserved? And they come back in. It's released as heat, wasted. And so then the electron transport train tries again to pump more protons out, but they come back in, releasing more heat. And then the electron train tries again, pumps them out, but they come back in and release heat. And how much ATP is being made? None. And then more fuel gets burned to make more high energy electrons. The electrons are pumped out, but they leak back in and they generate heat. And no ATP is being made, so more fuel is burned because you need ATP. Electrons pumped out, not electrons pumped out, hydrogen protons pumped out, but they leak back in. More heat is released. So instead of conserving the energy of fuel oxidation through oxidative phosphorylation into lots of ATPs, what are you making instead when you oxidize your fuel? You oxidize your fuel, the electron transport chain is working. Krebs cycle is working. Everything's working. You're burning your fuel like crazy, but instead of making ATP, what are you making? Heat. And what happens to the body? What happens to the cell? It's going to overheat. And it's not going to be able to make enough ATP to survive. And it's going to burn all its fuel trying to do oxidative phosphorylation, but because you uncoupled the chemiosmosis, okay, you're not making all the ATP that you should be making, and instead all you're making is a lot of excess heat. What do they use dinitrophenol for? One of its uses. That's a poison, R-E-O-R-E-S-O-N, right? Poison? Rat poison. Okay, so therefore, you know, if you have those little rats, once you have those little rats that they might be somehow in the wall of your apartment or whatever else it is, and they, you know, they come out at night and they're trying to eat, you know, whatever, they're going through all your food and they're going through your cabinets and they're chewing through everything, correct, trying to find food. Okay, so you have the exterminator come, and the exterminator lays out some dinitrophenol. And the rats, I guess, I don't know whether it's laced with something that the rats eat it. So the rats eat the dinitrophenol. And then what happens is they eat the dinitrophenol, they go back behind the wall, but sometimes they don't even go back behind the wall. And then suddenly what's happening? They're having uncoupling of oxidative phosphorylation, and they start burning up all their fuel, trying to make ATP energy. Because of the dinitrophenol, all they're generating when they burn their fuel is not ATP. They're just generating what? Heat, right? And then what happens usually with the rats is that because they're generating heat, their body temperature goes up and they start to get dehydrated. Most of the time, the exterminator will say, so then the rat will go looking for water because it's dehydrated and it becomes extremely thirsty. So that might mean that the next morning when you wake up, you might find a dead rat that actually jumped into your toilet bowl looking for water. Sometimes you just find him under the sink dead because he was trying to lick the pipes under the sink because they were wanting water so bad. Or better yet, sometimes if a rat got into an apartment building, it got in from the outside and it knows how to get out. So then it just goes out into the yard or whatever else looking for water out in the environment and just dies out there. Even if the rat died behind the wall, and we never want anything dying behind the wall, right? Even if the rat died behind the wall, because it actually overheats, it almost flattens out as a dehydrated rat. And dehydrated rats don't smell bad when they're decaying because you kind of need that water environment to decay a rat. So even if it died behind the wall, chances are it became like freeze-dried rat without any water and doesn't really smell that bad. So I always thought that was interesting, that story about dinitrophenol. I remember somebody who took the MCATs actually said that was one of the readings that they had on the MCATs, discussing about how something like that can happen in I guess in biochem, because it is a famous biochem story about something that interferes with chemiosmotic coupling and what happens. So they do use it in rat poison. And the funny thing is that some people said, how about a weight loss diet? Because think about this. If I take dinitrophenol, I will burn up my fuel, but I won't really be able to efficiently make all the ATP that I want. So I'll burn up more fuel. I'll get a little hot, but I won't take too much of it. And actually, this was something that was promoted back in the 50s, that the pharmaceutical companies were trying to make a therapeutic dose of dinitrophenol and make lots of money on it. Because wouldn't we love to eat all the food that we would want to eat? We wouldn't be able to store it. We would just burn it and would go off as heat energy instead of being stored as excess fat. Wouldn't this be the best thing, where you could have your cake, eat it too, and still lose weight? The trouble is, this is a type of chemical that the lethal toxic dose would be right next door to the therapeutic dose to actually get weight loss and not have you die. So the trouble is, it's never been useful as a way of actually eating all the food that you want, but just having the food burn off as heat. And you know how some people always seemed like they could eat everything and yet they never seem to get fat? And you're wondering, what are they doing with all that energy? A lot of times they probably are wasting a lot of the energy, meaning that they're not actually conserving it in a useful form, and that's why they can eat all that food. So this was almost like something that they were trying to think, maybe we can create this pill and stop this obesity problem. But the trouble is, they'll tell you like you could only take one pill a day. Just take one pill a day and you'll get nice slow weight loss release. You know what happens when you, and then all the warnings would be on the label, say, do not take any more than one pill a day, okay? Because this is potentially toxic. Okay? If you take too much, it's going to be very, very serious. And people go, yeah, I read that all the time. I take tons of Tylenol, I never get sick, right? No matter what the warning is, what do humans like to do? And if I took one pill a day, and after 2 weeks, and I didn't exercise or anything, 2 weeks, maybe I lost like 7 pounds in 2 weeks, and I didn't exercise, and I was eating the exact same way I always ate. And I was just maybe a little bit warmer, but I didn't really notice anything. I'd be so happy after 2 weeks that I lost 7 pounds, not exercising and not doing anything different. What would I then want to do? What would humans say that they would want to do, despite what the warning labels say? Well, if I could lose 7 pounds in 2 weeks, if I take one pill, hey, maybe I could take 2 or 3 pills a day and speed up this weight loss. And then what would happen to them? Just like the rats, they would probably what? They would probably die. And then, of course, what would happen to the company, the pharmaceutical company that made the pills then? They'd be sued, wouldn't they? Even though they have the warning label on, they'd be sued, correct? Because too many people would start dying. And many of the humans that would die, they would really die because they would not believe the warning labels. They would not believe it. They would say, oh, no, I've heard this before. And I'm a tough guy. I could do it, right? In other words, it's not going to kill me. So unfortunately, this is a chemical that they have tried over the years to try to see if they can formulate this thing or do something with this thing to try to make it a weight loss thing, because you make a lot of money on that. And all these companies, these pharmaceutical companies and stuff, all of them, every business wants to do this. They all want to make money, of course. And you can imagine the money that can be made, okay, as a result of having a pill that would make all the calories that you eat less effective at causing fat storage, and just have it burn it off, okay? And then you could just eat it and just burn it off and you wouldn't be getting fat. This would be great, wouldn't it? So far they've never actually come up with any kind of solution for this. And my final recommendation is, am I telling you now to go out and get rat poison and trying this? Am I telling you to go out now and take rat poison? It is called rat poison, right? Am I telling you to go out to look for this chemical and say, well, I'm going to take this? No, I don't want to, it's bad enough if my students fail the class, but I don't actually want you to die, correct? So again, and this is not secret information. You could see a lot of this stuff. You could read about this even on the internet if you wanted to. But I'm not advising anybody taking it. It is very lethal. There are people that have died of it, okay? And you don't see any reports of people successfully losing weight. They usually are dying before they actually lose weight because you can't control the level of toxicity. So it seems like it could never be a good drug as it is. But it's always interesting how it periodically comes up, okay? And you'll see it even on the internet if you want to read about it. Okay. All right. So I don't know. Let me see. What else do I have after this? Do I have something else after this? Okay, I guess we could answer this one. Mitochondria and oxidative phosphorylation. What do we call the process that involves the consumption of oxygen during chemiosmotic coupling reactions that also involve the phosphorylation of ADP? Well, that's what we call oxidative phosphorylation. Something gets phosphorylated, correct? And what's getting phosphorylated? ADP going to ATP, right? That gets phosphorylated. And while that's getting phosphorylated, what's being used up in the cell? Oxygen being used up in the cell, correct? Where does this process take place? In aerobic eukaryotic cells. That's like our cells. Where do we do oxidative phosphorylation? Don't just tell me mitochondria. Be more specific. I guess you weren't even going to tell me mitochondria. Where does this process of oxidative phosphorylation actually take place? What part of the mitochondria? Okay, good. The inner membrane of the mitochondria. Okay, here's another good question. Remember, mitochondria used to be what? Aerobic bacteria, correct? Right? Didn't mitochondria used to be aerobic bacteria that we engulfed and we kept? Yes, everybody knows that story, correct? All right, so now I'm asking you the question, okay, where does oxidative phosphorylation take place in an aerobic bacteria? But does it take place? Does aerobic bacteria, they do oxidative phosphorylation? Oh, Charmel got the answer. Is that because we're looking at the slide? I don't know. That was good. Okay. All right. Okay. Takes place in the plasma membrane. Most students will say it doesn't even happen because they'll go, bacteria don't have their own mitochondria. But remember, aerobic bacteria and mitochondria are really what? The same thing, correct? That's who the mitochondria used to be. Aerobic bacteria figured out the whole what? Figured out oxidative phosphorylation. And then us eukaryotic cells engulfed them. And now we have it because we have mitochondria. And when we think about the structure of the mitochondria versus the structure of the bacteria, all the bacteria has is the plasma membrane, right? Your bacteria, correct? And their little electron transport chain and their little what? ATP synthase is located there, okay? So they have an electron transport chain in their plasma membrane. Remember, if oxidative phosphorylation is a membrane-based mechanism and bacteria do it, the only membrane that a bacterial cell has is the plasma membrane. There is no other membrane in a bacteria. So any membrane-based mechanism that a bacteria does really has to be done where? In the plasma membrane. Okay? And of course, when this guy got engulfed, right, this probably came from, sorry about that. This probably came from the plasma membrane of the eukaryote who engulfed them, right? This membrane, the outer membrane, originated from the plasma membrane, like suppose this was a phagosome, right? So if you did like phagocytosis, this would have been the plasma membrane of the eukaryote who engulfed them. And then this used to be the plasma membrane of the bacteria. And of course, over evolution, what happened to that membrane? It got folded into a cristae, correct? But the origin of the inner membrane of the mitochondria is the plasma membrane of the bacteria that we engulfed. Say we did, that the eukaryotic cell engulfed. Okay, so where does oxidative phosphorylation, meaning where does the electron transport chain take place in an aerobic bacteria? The plasma membrane. Where is the enzyme ATP synthase in an aerobic bacteria? The plasma membrane, because that's the only membrane they have. And they invented this process. They invented the players of this process. Okay, you got it? All right. Which membrane is probably better at the process of oxidative phosphorylation? Which membrane do you think has evolved to be the best at oxidative phosphorylation? Do you think the plasma membrane of the aerobic bacteria or the inner membrane of the mitochondria in our cells? Which membrane do you think has evolved to be like an expert in doing oxidative phosphorylation? Tashira knows, right? It's going to definitely be the what? The inner membrane of the mitochondria, but why? Just even think about it. Here's my bacteria, okay? And here's my mitochondria. What if this used to be the plasma membrane? Remember that used to be the plasma membrane? What did we say happens to the inner membrane of the mitochondria? It got what? Folded, correct? It got folded. And what is the advantage of folding your membrane? You increase the surface area to do more work. And what kind of work does the inner membrane do? Oxidative phosphorylation work, correct? Notice that I can't fold the plasma membrane of the bacterial cell, can I? I can't fold that. It's a plasma membrane. But I can actually fold the inner membrane of the mitochondria, right? I can fold it. And if I fold it, I increase the surface area so that I could have more copies of the electron transport chain present. I could have more copies of ATP synthase present. Plus, the inner membrane of the mitochondria has become specialized. This is the only membrane the bacteria has. And that means this one and only membrane has to not only do oxidative phosphorylation, it has to do all the other activities of a plasma membrane. Whereas the inner membrane of the mitochondria, we can turn around and we can say to it, look, you're in a eukaryotic cell now. We'll do the plasma membrane stuff. You don't have to do that anymore. Why don't you just get really good at doing oxidative phosphorylation? Okay? That's the surface area, right? So you could see one of the effects is that through evolution that engulfed a bacteria, modified even the appearance of its so-called plasma membrane, which we now call the inner membrane of the mitochondria, through all that cristae folding. And in being able to do that increases the surface area and allowed the mitochondria to do more inner membrane work. What kind of work does the inner membrane do? It does a lot of oxidative phosphorylation. So it has the luxury of specializing. Okay? So it's specialized either in the structure of the membrane itself to get more surface area in a limited space. That's a smart thing to do. And it also specialized because now, through evolution, our mitochondria gave up a lot of the activities that membrane used to have to do when he was a free-living bacteria. Well, that used to be his plasma membrane. So there had to be all sorts of transport proteins to bring in, fuel molecules and all sorts of things that plasma membrane had to do. But now it doesn't have to do that anymore. That our eukaryotic cell says, oh, boy, mitochondria, boy, little bacteria engulfed. You really do something special that I can't do. You just get really good at doing that, and I'll take care of a lot of your other jobs that you used to do that I can do Because you can do something special. This membrane, the inner membrane of mitochondria, can do something special that no other membrane in the big eukaryotic cell knows how to do. So you just get really good at that and make lots of ATP for me through your oxidative phosphorylation. And I'll take care of you, and you don't have to worry about doing all that other stuff you had to do with your membrane when you were a free-living bacteria. Okay? All right. And let's see, mitochondria and aerobic bacteria are similar. I guess we can do this one too. Same general size and shape. They both have their own DNA and RNA. They both have their own transcriptional, translational system. Question? No? Okay. And with their own ribosomes. So these are how they are the same, right? These are some of the important ways that they're the same. Right? So how do they differ? They differ primarily because mitochondria are no longer autonomous. So we think about engulfing them and capturing them and making those poor little bacteria work for us and make all that ATP. And maybe they really want to be let free, right? Maybe they want to be free living bacteria. Maybe they don't want to live with us anymore. Maybe we should let them go. You know, maybe we captured them against their will and they're unhappy and they're just working for us. Well, through evolution, guess what happened? The mitochondria got so comfortable with the symbiotic relationship that it totally became very specialized for cellular respiration, oxidative phosphorylation. And if you let the mitochondria go, if you let him go, even though he's got his own DNA and RNA, okay, right? Even though he's got his own transcription, translational system, got its own ribosomes, if you let him go, he won't be able to survive on his own anymore. Okay? He's become so specialized to live with us, okay, that they say, go back to who you used to be. I'm going to free you. I'm going to release you. He's going to end up dying. In fact, through evolution, okay, our mitochondria only code for 37 genes. Okay? Only code for 37 genes, and all 37 of those genes are involved in oxidative phosphorylation, meaning they all code for genes that are associated with oxidative phosphorylation, something that they do. And many other genes that aerobic bacteria used to have in its DNA has been eliminated or lost through what? Mutations or loss, or possibly those genes have been transferred to the nuclear DNA. And we now have actually acquired those genes that used to be bacterial genes. We now actually have acquired them, and they're in our nuclear DNA, and we do all the transcription and translation for that. So they don't have enough genes to survive on their own anymore. They only got 37 genes in their mitochondrial DNA and are now totally dependent upon the eukaryotic genes and eukaryotic pathways for many of their needed proteins and other molecules. Okay, so I'm going to end there. I'm tired. All right, so I'm going to end there. I'm going to stop the recording. All right.